Welcome to the final video in this module where we have been discussing MHC. And in this final video, we are going to discuss something a bit more conceptual, and that is the evolutionary genetics of MHC. Why the particular way that the MHC is encoded in our genome, features like high degrees of polymorphism and polygenic encoding, why are those features evolutionarily advantageous, and how have they made populations more fit over time in the face of all of the evolutionary pressures exerted by pathogens. So to start thinking about some of the evolutionary implications of MHC, I'd like us to first sort of revisit the HLA complex, which, as we saw in the last video, is found on human chromosome 6. And here we can see those three regions that we were introduced to, MHC class 2, 3, and 1, as they are found going from centromere to telomere. And within those regions, we saw that there were many genes, including these classical MHC genes associated with MHC class 1 and 2. And a point that I want to make is that you can see that this region, it's a big region, but there's a lot of genes. Even though this is a big region, there's a lot of genes very close to each other. And so we think of this region as particularly gene dense. Genes are right next to each other, and there's not a lot of non-coding DNA in between. A implication of the density of the genes that are found in this region is that it discourages recombination during meiosis. So you may have learned before that one source of genetic diversity is the frequency of recombination as meiosis occurs, during gametogenesis. And during gametogenesis, in an individual, when they're making a sperm cell or an egg cell, the alleles on their own paternally and maternally inherited chromosome can sometimes swap places. And we're not going to go into the details of that in this video. If that's not something you're familiar with, I encourage you to look it up, and it may make some of what we discuss in this video a bit more easy to digest. But we're just going to start there by saying that meiotic recombination is incredibly rare within the HLA complex because recombination is sort of discouraged in regions that are dense with coding genes, because if you were recombining through those coding genes, that would be maladaptive. You would be losing those genes in essence. And there's some other features of this region of chromosome 6 which make it what we call a cold spot for meiotic recombination. Because recombination is rare, that means that these genes, which are all found next to each other, are linked and tend to be inherited together as a set. And they exhibit something that we call linkage disequilibrium. So linked genes that are inherited at random, meaning that any chromosome that is inherited sort of has an equal probability of having certain genes or certain alleles come from the maternal or the paternally inherited chromosome of the parent. We think of equilibrium there meaning a sort of random distribution of those alleles being inherited from one chromosome or the other, or from a gamete containing material from one chromosome or the other. Linkage disequilibrium, in contrast, the end result of it, again, for our purposes in this video, is that alleles tend to always be inherited as a set from either the maternal or the paternal chromosomes. All of them as a set tend to make their way together into any individual gamete. So that linkage disequilibrium tends to mean that whatever alleles are found on a given chromosome 6 within the HLA locus, they tend to stick together. And they stick together in something that we refer to as a haplotype. A haplotype we can simply define as the particular combination of MHC alleles that we find on any given single chromosome. And so every person then has two haplotypes of HLA, or MHC as it were. They have the haplotype represented on their paternally inherited chromosome 6 and the haplotype represented on their maternally inherited chromosome 6. And these haplotypes typically are very distinct from one another. As I said in the last video, most people are heterozygous for all of their MHC genes. So that suggests that the two haplotypes have to be highly divergent from one another. If we look at all of the alleles for all 9 classical MHC genes, remember there are 3 classical MHC class 1 genes, and then 6 classical MHC class II genes corresponding to all the alpha and beta chains of the 3 isotypes. If we looked at all of the alleles in all of the positions, most people are heterozygous, and so you would see 2 unique alleles represented between the 2 haplotypes. So what I'm showing you here are 2 kind of random example haplotypes. These are not real haplotypes that occur in nature, but what I want to emphasize is that all of the alleles that we could sort of list here that are associated with one chromosome, I sort of colored in blue as having coming from the paternal chromosome, and then a set of alleles coming from the maternal chromosome, I'm showing here in pink. Forgive the color choices. But in order to sort of understand this code here, which is sort of designates each of the alleles, I'll give you sort of a cheat sheet. I've simplified this coding just a little bit to make it simple for our purposes. But essentially, every allele that you might find in the human genome, there's an agreed upon nomenclature that we use to describe those alleles. It starts with the gene name, and that gene name corresponds, of course, to HLA-A, B, C, and so on. DPA would meaning the DP alpha chain. And then beyond that, alleles are sort of organized into genetically similar families known as allele groups. And there are many allele groups associated with each of the distinct MHC genes. And then within the set of related alleles or groups, the second number after this colon is something that identifies unique coding variants. And so we can really think of the second number as truly like the allele identifier of a unique genetic sequence, which defines an allele. And this first number sort of defines a family of related alleles that each individual member belongs to. So this is just a way to decode the sort of allele nomenclature if you encounter it. And you'll encounter even more complicated versions with additional numbers downstream. But I think this is the core information that you need to know. And so notice that, again, if I say that each of these alleles, this person is heterozygous, or you see that sort of played out here. For the HLA-A gene, there are 2 alleles represented, this 234 allele and this 344 allele. So these are 2 alleles that are actually in 2 distinct families, designated by 2 and 3. And within those families, these are the unique members for each. And so this person is heterozygous because we see that the alleles are different. And you can see that the alleles are different at every position, essentially. If we look at HLA-DR, you can see that this person expresses 2 different alleles, although they are closely related alleles because these alleles are in the same family, the family of 1. Nevertheless, even though they're in the same 1 family, they are 2 distinct coding variants, the 1 allele and the 3 allele. So the numbers tell us about how similar 2 alleles are and also which exact coding sequence is represented. But again, zooming out a little bit, I just want to say that if we define a haplotype, the first haplotype represented here is this unique combination of 9 very unique alleles, 1 for each gene. And then similarly, this is a haplotype represented by a distinct set of 9 alleles. So this person, because they have 2 distinct haplotypes, they will be heterozygous at each position. And of course, that will result in a different protein isoform being expressed for each of these unique genes as well. So another point to make about haplotypes in the context of inheritance is that haplotypes, because there is a low degree of recombination during meiosis, haplotypes tend to persist over evolutionary time. Because all of these alleles that you see represented here in blue, they will tend to always be inherited together and they will not be recombined into new haplotypes. That creates a very strong evolutionary pressure for any haplotype that exists to be a good one, to encode distinct alleles from A to B to C. You want all of these alleles to be different from each other because you want to increase the maximum amount of diversity that the haplotype itself sort of accounts for in terms of the peptides that can be presented. Similarly for the MHC class II genes. And so over time, haplotypes that contain alleles that are too similar to each other, they tend to confer a lower degree of fitness. People who have haplotypes which have low diversity within the haplotype tend to have less ability to fight infection, and therefore are less likely to survive and reproduce. So there's a selective advantage to haplotypes that have good coverage within the haplotype. That is one sort of feature of the fact, or one consequence of the fact, that haplotypes tend to stay intact even over subsequent inheritance events. So speaking of inheritance, we can sort of look at this a different way in one of these types of graphs. I've made the point over and over again that these haplotypes tend to remain linked across generations, and you can see that playing out here. If we take this particular mating event, and let's imagine that the person on the left here is the paternal parent, the person on the right is the maternal parent, each of these parents themselves have their own maternally and paternally inherited chromosomes. So in this paternal individual, you can see that they have their own paternal chromosome 6, which we'll designate as A, and their own maternal chromosome 6 designated as B. This maternal parent, of course, has their own maternal and paternal inheritance, and so we'll say that this dark blue is the paternally inherited chromosome for the mother in this scenario. The red is the paternally inherited chromosome for the mother. And as they mate and produce potential offspring, these are the 4 combinations of haplotypes that any offspring of this particular pairing could possibly contain, because there is a very low degree of recombination. What you can see here is that this potential offspring inherited both A and C. What this means is that this person encoded the haplotypes derived from both of their grandfathers on both sides, and they did not inherit any of the haplotypes that were represented in their grandmothers. This is a bit unusual because most genes have some degree of, or I should say most alleles have some degree of recombination from paternal and maternal sources as you go back through successive generations. But because of the high degree of linkage disequilibrium, you can see that these haplotypes remain stable over multiple inheritance events, as I said, and they tend to stick together. And so even though a potential offspring will have a different set of haplotypes, the haplotypes themselves remain intact over time. And so again, this creates a strong selective pressure for whatever allelic combinations exist within these haplotypes to work well together. And to get good coverage of the potential antigens that any individual that inherits that haplotype might encounter in their life. So if that's not completely intuitive, maybe let's look at it in a different way that may sort of make those points more conceptually again. So any given haplotype that a person might inherit might be a good one or it might be a bad one. So what defines a good haplotype versus a bad one? Or how we might say that as an evolutionarily adaptive haplotype versus a maladaptive one. So if we focus on MHC class 1, again, just because it's a little bit simple, more simple compared to MHC class 2, remember that there are 3 genes within MHC class 1, and that because we're talking about a haplotype, we're only talking about a single allele for each gene. We're on a single chromosome. If we think about the adaptiveness of a given haplotype, the way we might define that is to think about out of the set of total possible foreign antigens or non-self antigens that a person might need to present in their lifetime, if we imagine that set as the sort of gray circle here, and then we draw a Venn diagram of all of the possible antigens that any individual isoform of MHC class 1 might be compatible with presenting, you would get something like this. If the alleles that are present for HLA-A, B, and C are all different from one another, they're going to all have a different shape. And the more different from each other they are, the more difference in shape they will have, and therefore the more unique the set of antigens that each one will present will be. And so if you sort of imagine an antigenic space, which is what is being represented here by this gray circle, and within that antigenic space, there is a smaller space that is possible that can be covered or can be presented by each of these three HLA isoforms. The more different they are, the more of that antigenic space will be covered, meaning that this individual will be able to present a greater diversity, a greater coverage of that antigenic space, and that will be adaptive. They will be able to present more antigens and therefore be protected from more potential types of infection. In contrast, a maladaptive haplotype would mean that this person, even though they have three unique MHC class I genes, if the alleles for each are too genetically similar to each other, then the peptide binding groove of all three isoforms is going to be kind of similar, and it's going to bind a more overlapping set of potential antigens. And as you can see, the more overlapping these three isoforms are, that leaves more of the antigenic space uncovered, meaning that this person is going to be more susceptible to a greater number and a greater diversity of potential pathogenic threats. And so this would be evolutionarily maladaptive, because they are going to get sick more often with more types of pathogens. And probably that is going to decrease their ability to survive and reproduce. And so you can imagine that there's a very natural kind of selective pressure for haplotypes to contain alleles that are distinct from one another, versus those that are too similar. And because haplotypes have to be inherited as a set, that means that any haplotypes that have alleles that are too similar to each other are sort of weeded out. The evolutionary pressure selects against them over time. And so the haplotypes that we actually observe in nature, in human populations, tend to have sets of alleles that are more divergent from each other because those work better ultimately. So this is looking at a haplotype sort of on a single chromosome. But if we combine the idea that we just unpacked on this slide, and if we remember that we don't only have one chromosome, we have two, therefore we have two haplotypes, and we tend to have two different haplotypes. We tend to be heterozygous at all alleles. Then we get something that looks like this, where we get a combination of two haplotypes, and we most likely have two distinct haplotypes, meaning that for MHC class I, most people are going to have 6 distinct alleles, 2 haplotypes with 3 alleles each, all of which are dissimilar from one another. And what that means is that although many alleles will have some overlap with other alleles, they will all have some unique set of alleles that they are able to present. And that will give us the broadest possible coverage against all of the possible antigens that need to be presented. You can see here that the vast majority of this antigenic space is covered. And so this is the advantage of the combination of high degrees of polymorphism and codominant expression, because we can make actually 6 MHC class I isoforms, all of which are different from one another. We get the most possible protection. And I said that this is what occurs in most people because most human populations are what we refer to as outbred. Most people have very different MHC genetics from one another, and so when they breed, their offspring are going to most likely have 6 unique MHC class I alleles. This actually sort of tells us that one of the dangers of inbreeding is that inbreeding leads to homozygosity. And homozygosity for MHC genes is very maladaptive. And if we sort of try to graphically represent why that is, here you can see that you have 3 isoforms, but this individual is homozygous for the three MHC class I genes, A, B, and C. And so even though they still have 6 alleles, the sets of alleles within each isotype are redundant to each other. They're completely overlapping. And so even though 6 alleles are represented, you sort of only get 3 chances to cover space within the antigenic space that you see here. And so again, that's going to leave you more susceptible to any potential peptides that are not accounted for, that are not covered by the alleles that you see. And you can see that this person, the 2 haplotypes that they express, even though they're fully overlapping, the haplotypes within themselves are adaptive. The haplotypes within themselves still have a good bit of coverage. But because those 2 haplotypes completely overlap with each other, we don't get this full extent of coverage that you get in a person who is fully heterozygous at every position. And so, again, this sort of combines multiple features of MHC genetics to explain the evolutionary pressures that has made MHC function in the way that it does at the genetic level, because there are real evolutionary advantages to a high degree of polymorphism, and the fact that we have codominant expression has also been the result of a strong evolutionary pressure. And we can think of those evolutionary pressures with a couple of conceptual sort of explanations or organizing principles. And one of those is that the features that we see associated with MHC genetics tell us that it is under what we call balancing selection. Balancing selection is a type of natural selection that tends to maintain many multiple, distinct, diverse alleles within a population rather than favoring a single one. And it's actually more rare to be under balancing selection than to be under other types of selection, such as stabilizing selection, purifying selection. These are types of selection which tend to occur more often, which actually discourage multiple alleles and diverse alleles within a population, because it is sometimes more functional to have one good allele that functions all the time. And so this is very common, for example, for housekeeping genes, things like actin or certain metabolic enzymes, where you have one good allele and it serves its function, and if you mess with it too much, if you mutate it, it tends to be catastrophic. And so stabilizing selection for housekeeping genes tends to suppress the emergence of new alleles within the population. Instead, MHC shows a sort of opposite type of selection, balancing selection, where it's actually better to have more diversity, because on balance, even if one particular MHC allele mutates, maybe sometimes that'll be bad, but you've got a bunch of others to sort of account for it. But having those rare alleles sometimes comes in handy, as we'll see, because rare alleles, while they might not be useful today, might be useful in the future, because pathogens are constantly evolving and changing as well. So thinking about balancing selection, this type of selection that encourages the retention of multiple unique and sometimes rare alleles within a population, there are a few related concepts that sort of that flow out of the idea of balancing selection. One is that balancing selection is associated with the idea of heterozygote advantage, and heterozygote advantage is something that I was really explaining on the last slide, which is to say that individuals with two different MHC alleles for any given gene will have an advantage because of codominant expression. They are going to be able to present a wider variety of antigens, which of course is going to give an evolutionarily adaptive benefit. They'll be more protected from infection compared to a homozygous counterpart. I've made a lot of point on this slide to talk about the fact that balancing selection tends to preserve rare MHC alleles. This is a type of negative frequency dependent selection. Again, most of the time, for most genes, rare alleles tend to represent some mutation event, and mutations more often than not are maladaptive because they confer a loss of function. But this is less true for MHC, because again, you have backup MHCs that sort of help you even if one particular allele is maybe not as functional compared to the others that you have. But even though an allele might be dysfunctional in the context of a person's lifespan, they might sort of bequeath that allele to their offspring. And in the future, pathogens may have evolved in such a way that what was a less functional allele today will become functional in the face of new pathogen pressures in the future. So the retention of those rare alleles sort of increases the toolbox or the sort of the kit of possible MHCs that a person might ever express sort of over evolutionary time. And that tends to be adaptive again within the context of MHC, and we think of that as a type of frequency-dependent selection, the retention of rare alleles within the population. Finally, there's an idea called pathogen-mediated selection, which obviously is going to be very important for MHC, which is that different environments are going to have different pathogen loads, different pathogen environments, which are going to shape which alleles are most adaptive in any given geographical location and any given point in time, because across geography and across time, different pathogens are going to exist, and they are all going to have their own evolutionary pressures and histories. And so This is an explanation, for example, for why there is so much diversity of MHC across human populations across the globe. So it could be, for example, that yes, there's a lot of diversity within the MHC repertoire, but if you compared people of different ancestries, you wouldn't really see a very much difference. But that, in fact, that is not the case. Not only do we see a lot of population globally, but within individual populations with different geographical origins. So people with European ancestry or ancestry indigenous to the Americas or ancestries from East Asia, they have a lot of diversity within themselves, within those sort of inheritance groups. And then those groups are also diverse in comparison to other genetically distinct sort of inheritance backgrounds. That is because each of those populations historically has been evolving with a unique set of pathogenic pressures associated, again, with time and with place. And it's possible that this feature of MHC genetics actually may sort of diminish with current trends with globalization and the sort of intermixing of historically segregated populations. But at least as we stand right now, the MHC haplotypes that we associate or that we observe, again, in populations that descend from Europe versus East Asia, for example, there are unique haplotypes that actually are useful in some sense for allowing us to sort of understand the origins of people. And so MHC, because it is so unique, it's so polymorphic, and because it can haplotypes are so stable over evolutionary time, they're actually very useful for us to understand population genetics in terms of where individuals come from, how populations have migrated throughout evolutionary history, and so on. But that's a little bit of an aside. But one cool feature of MHC genetics that I actually think is it makes it useful even beyond the context of immunology. Okay, one final point about the evolutionary genetics of MHC is that MHC genes exhibit trans-species polymorphism. What this means is that we actually observe certain MHC alleles that we share with other non-human primates, and this suggests that they have persisted from our common ancestors. And this is also something that's a bit unique because many genes tend to be lost over speciation events. But the fact that alleles persist across speciation, meaning that there are certain MHC alleles which apparently are very useful and very good, very adaptive, and through balancing selection, they have been preserved over the course of millions of years. And in fact, this is also related to the idea of the retention of rare alleles because over the course of evolution, maybe these alleles at certain times were less useful and therefore they become more rare. But the more rare an allele becomes, actually there's a sort of inverse relationship with how useful it might be because it might confer at some point a unique advantage against pathogens as they are evolving. So over time, alleles tend to become more rare and then become become more common as they become more useful against pathogens, and then pathogens evolve in response. They evolve against those common alleles, common MHC alleles, and then they become more rare. But that is a sort of, an undulation, an ebb and flow that persists, again, over long periods of evolutionary time, and that's why we see alleles that sort of transcend speciation and go way back deep into evolutionary time. And so a point there is that the immune diversity that we observe in humans predates human speciation and is in fact something that is true basically for all vertebrates that have adaptive immune systems. So another interesting, I think, feature of MHC genetics that is a bit unique. Okay, summarizing this final video about MHC genetics, we see that MHC genes are inherited in discrete sets known as haplotypes, meaning the unique set of alleles found on one chromosome versus the other. So everybody has two unique haplotypes for MHC. I should note that I've said that a very high degree of linkage disequilibrium promotes the stabilization of haplotypes across inheritance. However, there are very rare recombination events that sometimes mix the haplotypes up, but that is very much the exception rather than the rule. We saw that selective pressures maintain especially adaptive haplotypes across evolutionary time. And what makes a haplotype adaptive is if it has divergent alleles within itself, within each haplotype, that provide broad antigenic coverage. And that is going to be evolutionarily advantageous against highly overlapping haplotypes. And when we combine this idea with the idea of polymorphism and codominant expression, these concepts together confer something that we call heterozygote advantage. And heterozygote advantage is one of the reasons that inbreeding is so maladaptive. Finally, we saw that MHC genes are under what we call balancing selection, a type of natural selection that favors the maintenance of diversity and high degrees of polymorphism in general, and that is certainly true within the HLA complex. One sort of consequence of balancing selection is that some allelic lineages are quite ancient indeed, and have been maintained across many speciation events, not only in human and primate evolution, but again, across many phyla within the vertebrate family tree. Okay, that covers it for MHC genetics. I'll see you in the next video, which at this point, I'm not sure when that will be, but at some point we will begin to cover the developmental origins of the immune system and look at lymphocyte development. Whenever that is, see you there.